stimulates other bacteria to do other things. In a biofilm, when sugar is around, not everybody knows that I'm going to even say this because I'm a fan of sugar. When sugar is around, it stimulates the bacteria to excrete a mucopolysaccharide. And this mucopolysaccharide is very, very sticky, which is what gets the bacteria to stick onto the tooth or whatever it's attaching onto. And this also is impenetrable. This covering cannot be penetrated by antibiotics. It cannot be penetrated by antibodies. And a lot of what we're using right now in dentistry, like mouthwashes like chlorhexidine and Listerine and all of these other things, have a super hard time penetrating that biofilm. You may get the surface up here, but you'll never get down in here. In order to use an antibiotic to get into a biofilm, you have to have 1,500 times the amount of antibiotic that you would if you were taking it internally. Or you would have to increase it to that level. And then what happens when you have 1,500 times the antibiotic that you normally would take? What happens to the human body? You get a stomachache? No. You assume room temperature. You die. You can't have that much. The other really cool thing about a biofilm is that the inhabitants of the biofilm have an entirely different genotype than the bacteria that's free-floating and commonly called planktonic bacteria. So once they're in a biofilm, they're a whole different germ. If we're studying, my favorite germ is strep mutans. If we're studying strep mutans in the environment, it's an entirely different germ when it's in a biofilm living together. I like to use this analogy. We've got mostly hygienists in here, am I right? Okay. So when you're a hygienist and you're at the mall, you act like a mall person. When you're a hygienist and you get together with a bunch of other hygienists, you act totally differently, am I right? Say yes. Yeah, you do. That's what happens to these germs. They have a total different way of acting and reacting once they're in that biofilm. They have things called DNA upgrading, where once there's some kind of a threat, they will change the DNA inside of them so that they're not threatened by whatever is threatening to that biofilm. So they're really safe. So these biofilms also react to fluid forces. There have to be fluid forces in order for a biofilm even to develop. It can't just be created in a dry environment. It needs to have some kind of fluid movement. And I almost said water, but that's not even true, because the earliest studies on biofilm were done on the pipeline, the oil pipeline that went from Alaska down into the United States. The biofilms would grow in the oil inside of that pipeline. So the fluid forces influence the structure of the biofilm, the shape of the biofilm, how many grow in how much area. It enhances the metabolic processes such as the genetic transfer. The cell collisions caused by the hydrodynamic forces lead to rapid spread of the genes, and that creates an evolution inside the biofilm that is super fast. They can create a super fast kind of an evolution because they're all jiggling around with their DNA, and they're squirting DNA, and their other germs around them take that DNA in and recreate themselves. Biofilms need the movement to survive. The genetic transfer may result in increased bacterial virulence and antibiotic resistance. Continuous exchange of DNA, bacteria are constantly evolving and communicating by the gene transfer on top of it. We have this signaling going on where the bacteria produce hundreds of proteins that the free-floating bacteria do not. If you remember the studies that we've done in our own educational experience, we would take some schmutz off of somebody's teeth or our own, and we'd smear it onto an agar plate that was intended to grow out one particular germ out of that mass, am I right? So that's how we've always studied these germs, but they act differently in a biofilm, so we're really rethinking a lot of what we've always thought we understood about a particular bacteria. They also communicate cell to cell individually, and some of the proteins are signaling molecules that trigger more genetic activity, and the genetic activation creates bacterial aggregation, matrix formation, and adhesion. Here's a cells-to-cell communication, and you can see we have some green cells here, some green bacteria, and they're talking to the red stuff. We have the green ones in this tower over here talking to the mushroom inhabitants. So this would be like a long-distance phone call from Chicago to Lafayette. And what else is on Houston? Houston, we have a problem. So anyway, we have all of this kind of communication going within the biofilm and outside of the biofilm. So this could, all of these germs right here, all of these biofilms, all of these little shapes could all be in one tiny little speck on your tooth in the pocket. We have microelectrodes that give us so much information. So if you think a biofilm is very, very small, I don't know, pick a small number. Now you take a little electrode here that's 1/1000th of an inch, which is about the size of a hair, and you put it into that biofilm here, you will get a temperature, a pH, and an oxidizing or an oxygen level of one kind of a number. And if you move that probe over within the same biofilm over just a tiny bit, you can have an entirely new list of markers. You could have a totally different oxygen concentration, a totally different temperature, a totally different pH in the same biofilm. Metabolic activity can change between different sections of their own biofilm. It's a heterogeneous mass. It's not just one single germ overgrowing into some kind of a white crud, on people's teeth. Bacteria survive in widely varying oral conditions. You can drink a whole can of Mountain Dew and still not kill a biofilm. It's true. This is a picture of the different ways of measuring and where they can measure in between the biofilm or into the biofilm. It has this matrix shield to protect it from everything. When this information first came around, I was in a lecture with Dr. William Costerton, who is like the godfather of biofilms, and he started this whole big biofilm studying school in Montana. He's now shifted his research to be all dental, and he is now at the dental school in Southern California. And he showed us a video of this incredible amount of white blood cells trying to penetrate through this matrix. And all of these white blood cells are like quivering around this biofilm, and they can't get in. And what happens with a white blood cell, if you remember, it creates this lysing fluid that's supposed to break down the cell walls so that it can kill the bacteria. Well, it never can make it in there, and that lysing enzyme can spill all down around that biofilm, and it'll break down whatever is in the way. It'll break down, tissue, it'll break down bone, it'll break down a bunch of other things, but it won't penetrate into that biofilm. It's the beta-lactamase enzymes are not penetrating that matrix. It's produced by the bacteria, degrade antibodies faster than they can diffuse inward, and dominant bacteria live in the center of this biofilm. Antibiotics, if you remember, work on replicating cells. I've got a little quote here. The bacteria is found, that replicating bacteria is found on the outer part of these biofilms. And if by chance some of that antibiotic can get in, it will work really well on the outer surface of that biofilm if it can penetrate. But as we get nearer and nearer the center of the biofilm, there's no replication going on, and there's a lot of dormancy. The penicillin works on cell wall synthesis interruption. And as this, let me see the next slide. Let's pretend we have an antibiotic that can penetrate into part of this biofilm right here. We have an antibiotic kill zone that may or may not work here on the outside and get maybe down into here where all of this is dormant. Then it's done. And then these guys get reactivated. Whoever's living in there dormant. So you can't really kill a biofilm. And it's not just a periodontal disease kind of a biofilm. We have a lot of trouble with prosthesis, like hip joints and knee joints, and wherever else you would have a prosthetic, where the biofilm starts before it's entered into the body, and then it just starts to grow and grow and grow, and they can't kill it. You can't kill these biofilms. They have to take, let's say if it's a knee, they have to take the knee out and let the person just rest in bed with no knee until that infection gets under control. I heard an awesome little research project. I heard about an awesome research project last week that I'll tell you about as we get going to help get rid of these kind of infections. These diabetic feet infections are also biofilm infections that are almost impossible to get rid of, which is why they have to amputate so far up in order to get rid of those infections. We also have a thing called persisters. So here we have, this is a diagram of biofilm tolerance. We have an antimicrobial agent here that may or may not penetrate into this first little part. We have gene transfers going on over here because these bacteria are perceiving an onslaught. They're perceiving some kind of a danger, so they start swapping out their genes. Now, we have a whole novel phenotype growing down in here, and the replication is very, very slow. But see these little purple guys in here? These are persisters. They will always be here. They are almost impenetrable. You can't kill those with anything. They just keep shifting and shifting and shifting until the danger is past, and then they explode, and it takes almost no time at all to rebuild that biofilm from whatever it is that we are able to get into to get a level of change here. This is a little study that I just found the other day. And I wanted to put it in here because it's a cool word that we don't really think about very often when we're talking about bacteria, the sociobiology of biofilm. Isn't that a cool word? Don't you just love Latin? You can just mix and match all day long. It's like our animals. Many bacterial species form the biofilms, but what they're finding is that it is a multicellular organism. So the biofilm itself is an organism. I can't say that enough. And that they have a sociobiology going on in there, and they have a social evolution inside of this biofilm. And they had two different conclusions out of the study. The first was that this biofilm properties such as phenotypic differentiation, species stratification, and channel formation don't necessarily require that cells communicate with one another. This is an ongoing science. This is the first study I saw that said that the form sensing isn't as important as what we originally thought. And that the biofilm has a competition and a cooperation between the bacteria living in there so that there's all kinds of promotions. So now we're trying to figure out how we can get rid of these biofilms in the easiest way. We can brush and floss it, but that only works if you can reach it with the brush and floss. Am I right? What if you have a 6mm pocket? You can stand as close as you want to that toothbrush. It's not going to help. Okay? Not all bacteria are equally capable of causing disease. The early colonizers are beneficial, like Actinomyces and Streptococcus. The later colonizers, this group down here that we don't ever talk about in polite society, those are the pathogens. They come much later. the bacteria attaches onto something, either the tooth or the pocket epithelium or other attached bacteria.